So, Kingdom Plantae, all plants have something called a cuticle. So, the cuticle is a waxy protective coating. So, if you recall, we talked about waxes being lipids and how lipids have a protective function. So, anyway, that's the shiny part on like a leaf. Like if you look at a plant, you'll notice usually the top side is shiny and the bottom side is not. So that cuticle is a wax, it's a lipid, and it protects the plant from water loss from evaporation from the sun. That's usually why the top side or the side the sun hits is shiny and the bottom side is not always shiny. Anyway, so all plants, some characteristics, all plants are made of one or more cells. Okay, so that means they're multicellular. Okay, they're also eukaryotic, so they have a nucleus and the membrane-bound organelles. They're also all autotrophs. Okay, remember autotrophs are also called producers. They produce their own food through photosynthesis. Okay, and remember that's in the chloroplast. The chlorophyll absorbs the sunlight to help them make sugar. Plants have cell walls made up of the sugar, the polysaccharide cellulose. So remember, cellulose is a polysaccharide, a mini sugar. Okay, so that's a carbohydrate. Now, you know how we say kingdom, phylum, class, order, family, genus, species? Well, instead of phylum, we have kingdom division. Okay, for plants. This is only for plants. And there are 12 divisions of plants. Now, a botanist is someone who studies plants. If you want to learn more about plants, you could take the class botany. Okay, so plant reproduction. So plants are grouped as either vascular or non-vascular. So they either have vascular tissue or they don't. Vascular tissue is the xylem and phloem. Okay, so you can think of vascular tissue in plants as like veins, okay, in plants. Okay, so if you ever see a leaf, so if here's the leaf, you know I'm not an artist. But anyway, here's a leaf, and it'll have the little lines, and it'll kind of look like this. Okay, or if you go outside and you pick a blade of grass, usually grass is going to be thin. So if this is grass and they have parallel lines. Anyway, so that's vascular tissue, those lines. It's just like your veins in your body. It transports blood. Well, plants don't have blood, but this vascular tissue does transport water. So the xylem transports water, and the phloem transports the food in the plants. So anyway, vascular, non-vascular. Non-vascular plants are usually very small because they do not have vascular tissue to transport water and food long distances, so they're very small. And they must have water to reproduce, okay? Now, they have what we call spores, or basically they're sex cells, and they have either sperm or egg in them. And they have to travel to water in order to for the sperm and egg to come together. So they have to have water to allow the sperm and egg to come together, and also so that they don't dry out. Okay, germination is very similar to fertilization. Not exactly the same, but it is when the egg and sperm come together. So very similar. Alternation of generations is something that some plants go through. It's 2 stages, where one stage makes a haploid cell and one stage makes a diploid cell. And this usually depends on the conditions, if this plant has favorable or not favorable conditions. So the gametophyte stage, gametes are haploid, so the gametophyte stage is going to make haploid cells. The sporophyte stage is going to make diploid cells. Okay, so this is going to be still produced during meiosis. So gametophytes are the only plants that have a dominant gametophyte phase. All the other ones are mostly going to be in the sporophyte stage, unless they're put in like poor conditions, like not a lot of water. Okay, so here's just a closer up picture of how, if you look at the top, the sporophyte is made, and it's diploid, and the bottom the gametophyte's made, and it is haploid. And it does require the sperm and the egg, the two different gametes, to combine for fertilization to make the zygote. So vascular plants are much different than non-vascular. Remember I said non-vascular plants don't have that vascular tissue to transport the food and water long distances, so they're very small. Vascular plants can be any shape or size. They can reproduce with or without seeds. So vascular plants are classified by either having or not having seeds, and the seeds are for reproduction. So your seedless vascular plants do not make flowers. Okay, they do not make flowers, they do not make seeds, but they reproduce using spores also. And this is usually going to be in warm, moist climates, very tropical type climates. Seed vascular plants, so these are seed-producing vascular plants, are then further divided into gymnosperms and angiosperms. Okay, so first off you have vascular and non-vascular. Okay, and then, so your non-vascular, they just are non-vascular. So then your vascular plants are further grouped into seed or seedless, and then your seed vascular plants are further grouped into gymnosperms and angiosperms. And so your gymnosperms are going to have seeds in a cone, and the cone is considered a naked version of the seed. Okay, and they do not produce flowers. So think like you've seen these things. We have those little palms, like little short palms are called sago palms, and you'll notice they have the big cone in the center. Anyway, so seed-producing gymnosperms have cones, and that's considered a naked version of the seed. Then you have your seed-producing angiosperms. Okay, angiosperms are flowers, and there's only one division of flowering plants out of the 12. Now, flowers are not always big and beautiful and colorful. A lot of the times they're not even noticeable. But the flower is a covering for the seed. Okay, so angiosperms have covered seeds. Gymnosperms have naked seeds in cones. Now, angiosperms are the only division or only type of plant that has all four organs of the plant. They have the root, stem, leaf, and the flower. So they do produce seeds inside of the flower. Now, those seeds will develop into fruit. The fruit, well, not always, but some seeds can develop into fruit. And fruits are actually considered like the matured flowered part, matured ovary. Okay, so you'll get the flower first, then you'll get your fruit or even your vegetable. Anyway, so another thing I wanted to say is about flowers not being big and beautiful. Grass is a flower. Okay, a lot of people don't realize that, but grass is a type of angiosperm. Okay, so that brings us to kingdom Animalia. And all this is very general overviews. But anyway, all animals are multicellular, and they have no cell wall, and they have no chlorophyll. They're all eukaryotic and heterotrophs. Remember, heterotrophs are consumers, so they must consume something to gain energy. So they do not depend on other organisms for energy. Now, one thing about animals is that they are all capable of movement at some point in their lives. The movement helps them get food, find protection, find habitat, and also, of course, to reproduce and find a mate. So here are some animals. So, some animals have specialized cells that can form tissues, organs, and organ systems. Now, animals are classified in a couple of different ways. One way is having structural similarities. For example, primates all have opposable thumbs, so you are a primate. Evolutionary relationships. So how things evolved, if they believe they came from a more common ancestor. So all birds are classified together, for example. Another way that they're classified is by body plans. Okay, so you do need to recognize these body plans, and I'll show you a picture in a second. Asymmetrical, A is without. Okay, we know A, the prefix, means without. So it's without symmetry. Okay, so they do not have a specific shape. And the only thing we really usually say for that is a sponge, like a sea sponge. Then you have radial symmetry. Okay, so if you take the radius, what do you take the radius of in math? It should be a circle, right? Okay, so a radial symmetry animal is going to be around a central point. Think like a pizza is a circle. Sea stars are circular. Sea anemones and things like that are circular. And then you have bilateral symmetry. Most animals are going to be bilateral symmetry. So if you cut it down the middle, it will have two equal halves. And all vertebrates are going to be bilateral symmetry. Okay, so here is an animal cell, and we've learned all these parts before. Okay, and here's our symmetry. So if you notice here with asymmetry, it doesn't matter how you cut it, it's just all over the place. It's very different. It doesn't have a specific shape that you can define. Radial symmetry, you can see it's around that central point. Bilateral symmetry, again, you can cut it right down the center. Sorry, I'm not a good drawer, but if you cut it right down the center, you should have equal halves. So you have 9 phyla of animals. Remember, your kingdom is Animalia, and your phyla is one of these things. Okay, so porifera are sponges, things with pores. Okay, Cnidaria are things that sting, like jellyfish and sea anemones and hydra. Platyhelminths are your worms, or your flat, specifically they're flat worms, okay, like a tapeworm or a, that's probably the main one that y'all would know. Nematoda are round worms, okay, so like hookworms or pinworms, and those are like the worms that your dog might get, and that's why you give them the worming medication, which sometimes the worm medication's a pill, sometimes it's like stuff you put on their back. Mollusca are like mollusk. They are soft-bodied animals, like octopus, clams, oysters, snails, things like that. They're soft-bodied. They may or may not have a shell. Squid. Annelid means rings. Okay, so these are segmented worms like earthworms. Leeches are an annelid. Arthropoda are going to be things that are going to crunch usually. So this includes arachnids, which are your spiders, crustaceans, which would be like crabs and shrimp and things with shells that are not going to be mollusks, lobsters, and then this also includes insects. Arthropoda includes insects. So as you can imagine, Arthropoda is going to be your largest phylum because of the insects. But anyway, your 8th phylum is Echinodermata. Echinoderm means spiny skin. So these are like kind of bumpy sea creatures, sea stars, sand dollars, things like that. Okay, and then Chordata, that's where we are. Okay, so all your birds, all your mammals, reptiles, amphibians, things with bones usually, and they are vertebrates. Okay, so Chordata that's what you are. So this is just showing kind of a branching type pattern of how animals are, how the kingdoms have been broken down and how animals and plants are broken down. So you have where they believe life began, sorry. So then you have your eubacteria, your archaebacteria, this is protists, then you have fungi, and then you get into your plants, and then your animals. Okay, so you have your very simple animals like your sponges, your cnidarians, flatworms, and then we get into more complex like roundworms and arthropods. And anyway, you can look at it further, how they've got it branched out and evolved. All the following kingdoms include autotrophic organisms except. So what does not include autotrophs? Hopefully you know animals are not autotrophs. Which of the following is not considered a living organism? Hopefully you recall viruses are not alive. Parameciums are protists. Mushrooms are fungi. And bacteria, nitrogen-fixing bacteria are specifically eubacteria. But if you weren't sure, it could be either eubacteria or archaebacteria. Either way, we know bacteria are living. Okay, what kingdom do these characteristics go with? Unicellular, prokaryotic. They may be aquatic or terrestrial. Remember, aquatic means living in the water, terrestrial, living on land. They cause disease, and they're used in food and medicine. Well, hopefully you realize that prokaryotic, the only example is bacteria. And there's only two bacteria, either archaebacteria or eubacteria. If they cause disease and are used in foods and medicines, they're true bacteria. So they're eubacteria. Okay, algae, slime molds, flagellates, ciliates, sporozoans, and amoebas. What kingdom is this? Hopefully you recall, this is Protista or Protist. Okay, unicellular prokaryotes in a harsh environment. So prokaryotes, bacteria, right? What kind of bacteria? Eubacteria or archaea? Harsh environment. Hopefully you picked archaea bacteria. Mushrooms, mold, puffballs, mildew, yeast. What is that? That is fungi. Ancient prokaryotes. Again, prokaryotes. Bacteria. Okay, ancient bacteria. Hopefully you picked archaebacteria. Okay, most are unicellular, but they're eukaryotic. They may be aquatic or living in moist habitats, and they may be auto or heterotrophs. Okay, hopefully you picked Protista. Remember, Protista typically live in water. True prokaryotes. We know that true means is you, and prokaryotes are always bacteria. Multicellular or unicellular, but they're eukaryotes and they absorb nutrients as decomposers. Okay, so what is that? Fungi. So speciation, we've talked about several times now, is the formation of a new species. And then a common ancestor, of course, that's the parent species where two or many species evolve from. We've used both of those terms several times now. So one thing we're going to talk about now is phylogenetic trees or phylogenetic trees. It's the same thing if you see it spelled a little differently. These are just a diagram that can can be used to represent evolutionary relationships, okay? So what they do is they can show how one single cell evolved into all the kingdoms we have today. Another thing they can do is they can trace how divergent evolution occurred and when at what point they believe speciation occurs to create a new species. So common ancestors are represented at each split or each branch on the tree, and then each dot or what we call nodes is representing a common ancestor for any organism evolved after that split. And so as time progresses forward from the trunk to the branches, the trunk may or may not be at the bottom, so it might not look like a traditional tree. Anyway, the length of how long the branch is or the line, that usually indicates how long of a period of time has passed. So a shorter line, a shorter branch. So a shorter branch is going to indicate a shorter time period, and a longer branch is going to indicate a longer time period. So if you look here, this would be our common ancestor, and that's represented here. Okay, and then E came off of the common ancestor. So the species E has branched directly off from the common ancestor. But then all these others have diverged. So this line represents a long period of time. Okay, so then off of the common ancestor, so over a period of time, they diverged. Okay, and then you got species A and B over here that are going to be related, and then species C and D over here that will be related because they're coming off of the same line here. Okay, so this is another picture. It's a much larger picture. So this one shows multiple kingdoms here. Okay, so you have your animals, your fungi, your plants, and so down here we have our common ancestor. Okay, so that's where they believed everything would have come from. And then we have our bacteria, which would be like archaebacteria and eubacteria. So that would have come off of the common ancestor. So that's what they're thinking was the single cell, and bacteria are single-celled organisms. And then we had our mitochondria and nucleus come up. And so all these things are going to be eukaryotic. Okay, so we have our chloroplast, and so all the plants will have the chloroplast. And then you have different things that come up, like the vascular tissue, the seeds, enclosed leaves, that would be flowers. And then you have your fungi branching off, and then you have your simple animals branching off, then animals with organs branching off, and then organ systems branching off. Okay, and so all these things show up, okay? So everything after this point, so if you look at vertebrae here, everything after this point, okay, so all these things are going to have vertebrae, okay? But if you look here, here's feathers, okay? Only birds have feathers. So, and this is just one more example of a phylogenetic tree, okay? So if you look, a lot of the time, these do have all the kingdoms on them, okay? So down here, starting at the bottom, you have archaebacteria branching off from our common ancestor, and then we have all the other bacteria branching off, and then chloroplast and mitochondria would have showed up after these bacteria, right? Because everything else other than bacteria are eukaryotic. So then you have your protist coming off, okay? Your diatoms, your algae, and all your algae, and then, so these would be like, hang on. So these would be some protist, and then these would be protists that were probably photosynthetic. And then after that point, they became plants. See, those are all your plants. And then up here, yeast, there's your unicellular fungi, and then your multicellular fungi. And then we come over here, and we get more protozoans. So those are more animal-like protozoans and fungus-like protozoans. They're heterotrophs. And then you have your animals coming off of this branch here, and vertebrates at the very top. So in this phylogenetic tree, this shows all these different bacteria, so archaebacteria, eubacteria, and then you branch off to all your eukaryotic things, which is going to include plants, fungi, animals, and then all these things are going to be, other things are going to be protists, which are going to be animal-like, fungus-like, or plant-like. So a cladogram is another diagram that we can use to study evolution and to show relationships between organisms based on their similarities, based on their adaptations as they evolved and characteristics that they received as they evolved. So a cladogram is unique because it'll show key evolutionary characteristics that separate one group from another group. For example, birds all have feathers, so it'll show that characteristics, the feathers, that make birds different from other organisms on the cladogram. So those things, those characteristics, or those adaptations are called derived characters. Okay, so a derived character is going to be found in the new species. So where you see the derived character on the cladogram, it will show up in everything evolving after that, but not before that. So it's going to be absent in previous lineage. And when studying a cladogram, time is represented as going from the lowest point of the V to the highest point. So you would begin reading it from the bottom node to determine, which direction you should start in. Branches that are going to occur sooner in the timeline are going to represent organisms that evolved before others. And those derived characters were those characteristics like feathers in birds, or having a jaw in fish, laying eggs. Anyway, so once it shows up, everything past it, everything that evolves after it will have that characteristic. And I'll show you on a picture in just a second. So any before it will not have it. So here is an example of a cladogram. Okay, so if you're looking at this cladogram, this shows animals. Okay, and if we start here, okay, so this is where it would begin. I would start reading here at the first little V. Okay, so anyway, we have the hagfish which branched off first. Okay, so then you have jaws show up. So notice jaws showed up after the hagfish. So the hagfish will not have jaws, but everything after the hagfish from this point on, everything from this point on branching off will have jaws. Okay, so that's what I meant. So the jaws is the derived character. Okay. That's what I was trying to say. I'm trying to clean that up a little bit. So then you have lungs. So everything before this, the hagfish and the perch would not have lungs, and everything after it, the salamander, the lizard, the pigeon, the mouse, the chimp would have lungs. Now another thing I want to point out is here, feathers. Okay, so notice this is on this line. So only the things past this point are going to have feathers. So only the pigeon has feathers. Okay, the mouse and the chimp did not have feathers because it was not on the line that they share. Okay, but the mouse and the chimp will have fur and mammary glands, whereas the pigeon, the lizard, the salamander, the perch, the hagfish would not. Another question that you might be asked on this is which two organisms are closer related? So I might say, what is a chimp closer related to, a mouse or a pigeon? Well, because it's closer to the mouse, it's closer related to the mouse than the pigeon. You have further on the line to go to get to the pigeon. So what is the salamander here closer related to? A hagfish or a lizard? Okay, well, it's going to be closer to the lizard than the hagfish. So the salamander will be closer related to the lizard. So that's another type question you could possibly see about a cladogram. So this is a diagram that's going to show how new adaptations, like these derived characters, how they show up in organisms as they evolve. So here's another example of a cladogram. Let me fix that a little bit. Okay, so we have our derived character, the vertebrae. Okay, so that was the adaptation. And so everything after that will have a vertebrae. Okay, so you have sharks. Now sharks have vertebrae, but they have a cartilage skeleton. Okay, so after the sharks, bony skeletons come up. So sharks do not have a bony skeleton, but all these things do. Okay, so anyway, and if you look here, we have forelimbs is another adaptation, an amniotic egg or eggs with shells. Okay, so here all these things have amniotic eggs, but these do not have shells. These do. So yet another cladogram. So this one is going to be dinosaurs. How they believe dinosaurs have branched off. So we would start out with our archosaurs, and then you have your crocodiles and their relatives, and then your ancestors related to birds, your pterosaurs, and then you have actual dinosaurs. So these would all have something in common, and then these and these, they're going to evolve. Okay, this is an excellent picture because it combines genetics with evolutionary divergence and how things have evolved with the cladogram. Now the cladogram you see is in white here. Can you see the cladogram there? Okay, so there's your cladogram. Okay, but they've overlaid it with a graph. Okay, so the graph is the number of amino acid differences between the hemoglobin polypeptide and a human one. So if you look at the bottom of the cladogram, we have the lamprey. Okay, so the lamprey is going to have pretty much 125 differences compared to the frog. Okay, so if you look, there's a big gap here between the lamprey and the frog. Okay, so that's a big amount of differences if you did that math. Okay, but then between 45 and 67, between the bird and the frog, you're going to have a smaller gap. So, anyway, and then the human and the macaque, this is a much smaller gap, only 8 differences. Okay, so this kind of shows you that how big the lines are is relative to the number of differences you see. So again, I could ask you, what is closer related to the dog, the bird or the macaque? Okay, so the bird is going to have 45 differences. So here's the bird, and here's the dog. Okay, and so the dog, the line is closer between the bird and the dog, and if you did the math, 45 minus 32 is 13. Okay, and then 32 minus 8 is going to be 24. Okay, and you can see the length of this line is going to reflect that difference. Okay, so that brings us to evidence of evolution. Okay, so how do scientists believe, how do they figure out these things and how things have changed over time? So these are the things we use for evidence of evolution. Fossils, okay, fossils are really one of the only ways we can see into the past. So fossils are very important as an evidence of evolution. So looking at fossil records, comparative anatomy, okay, so comparing structures. Okay, so these are structural similarities. We also can compare embryos. Okay, embryos, remember, are before birth. That is what this picture is pointing to. And then this is a fossil, and this is comparative anatomy. We also can compare DNA, and then we can compare the biochemistry, okay, of things and see similarities. For example, we can compare different proteins that different animals have. Okay, so if you take a look at these embryos, we're comparing a fish, salamander, tortoise, chicken, pig, cow, rabbit, and a human. Okay, so if you look up here, they look very similar. Okay, now as you get down here, they're going to begin looking more and more different. But because they are similar here in early development, that makes people think that they've come from a common ancestor. Same thing here. It's a great picture of homologous structures. The purple bone is the same bone in the human, the cat, and the whale. The hot pink bones are the same bone. The different color yellow bones, this picture is showing you that a human, a cat, and a whale all have the same bones. They might be a little bit shorter or longer than the others, but because they all have the same structure, the same bone, that makes scientists believe they came from a similar common ancestor. So, scientists have identified about 2 million different species. So this is like they've named about 2 million different species. And this number is constantly changing because they're constantly naming more. They believe between 5 and 100 million species have existed or currently exist. Extinct mean they no longer are living species. Extant is a still existing or still living non-extinct species. Scientists believe more than 99% of all species that have ever existed are now extinct. So, scientists, like I said, have used these 5 methods to study evolution. And so they can observe how species have evolved over time through fossils, embryological development, those structural similarities, how they're made up, the physiology, and DNA comparisons. And so most scientists today consider similarities in DNA to be the best indicator of how closely 2 species are related. Okay, so this should make sense to you, because if I need to see if 2 organisms are related, so if I need to see if 2 humans are related, if a child belongs to a father, or if 2 children are brother and sister, then DNA is going to be how I would do that. So it makes sense that I could compare the DNA between a cat and a dog to see the similarities in the DNA. Okay, so DNA is going to be the best way to conclusively see how closely 2 things are related. Okay, so here you have the evolution of what they believe to be a horse. So if you pay attention, you're looking at the foot here and how it's evolved into the current horse with hooves. Okay. And they've gotten much larger too. And if you look, this would have been 80 million years ago all the way to 3 million years ago. So this is evolving over a long period of time. Okay, so like I said, the fossil record is one example or one evidence of evolution. Okay, so again, you have the horse here. It's a little bit better picture. Evolving from a very small organism to a much larger organism with hooves. And those hooves do help support the higher weight. Anyway, so the fossil record, that's the first evidence of evolution. We know that fossils can show evolutionary changes over time, and this is really the only way we can see past living things. And so fossils are found usually in sedimentary rock. And what a fossil actually is the preserved remains of part of an organism or the whole organism. And sometimes it can be like a footprint, or it could be a bone or a leaf. So it might not be the whole organism, but it might be just part of it. So anyway, fossils, how they form. Well, not everything that dies becomes a fossil. A lot of things that die, they get eaten by something else, or they decompose naturally. But if they are given certain conditions, a fossil will form. So how a fossil, and fossils are going to be more likely to form in certain areas. So they believe that, a lot of areas, we might not have been able to see what lived there, and or they got eaten. So fossil formation, shortly after the animal dies, before it's too decomposed, they will be completely covered in mud or clay, and that mud gets compressed over time, and it gets layered into the rock layers. So more and more and more mud's going to go on top of it, which means more and more and more weight will go on top of it, and it eventually compresses it and pushes it down, and it layers into the sedimentary rock. And then over time, different minerals are going to replace, if it's a plant, the wood or the shells of the organism, or even bone. And then it becomes either petrified or fossilized. And then we can see kind of what was going on back in the day. So ooh, y'all can't see that, I'm sorry. Or maybe you can. So a paleontologist is a scientist who studies fossils, specifically found in sedimentary rocks, and this is usually going to be found in shale, limestone, sandstone, and conglomerate rock. And they have found that specific species only live during specific time periods based on those rock layers. So they look at the rock layers. Geologists are going to study the earth and the rock layers. Okay, so they can date the rock layers based on where they are. And kind of figure out how old the rock is, because the oldest rocks are going to be on the bottom and the youngest rocks are going to be on the top. Okay, and if it's in the same layer, then it's the same age, generally. So strata are the layers of rock. So anyway, paleontologists, they're going to look at the rocks, the fossils in the rock, and it kind of helps them age. And they've found, like, dinosaurs, for example, lived during a specific time period. So there's a couple ways we can date things. So one way is radiometric dating, and the other way is relative dating, to determine kind of the time period the organism lived, and neither is like just super accurate. So relative dating is basically looking at the rock layers, and a deeper rock layer is going to be older. And so I can know that if an organism is found in a young rock layer closer to the surface, then it'll be more recent years, and then the further down, it'll be older. Index fossils are organisms that existed for a very specific time period before becoming extinct. But when they did live, they were very widespread, living in many parts of the world. One example of an index fossil is called ammonites. They were mollusks, so they lived in the water. They were soft-bodied organisms with shells, and they lived during the Mesozoic era, which, was a very long time, million years ago. MYA is million years ago. And they were in pretty much all the oceans worldwide. So that would be an index fossil because they were very widespread all over the world. But they are extinct now and lived during that specific time period. So we saw lots of them during that time period, and then they just disappeared. Okay, so this is showing you relative dating. Okay, so if you look to the side, they've given you a key of what the different rock layers are. Okay, so your youngest rocks are going to be up here, up at the top. Okay, and your oldest rocks are going to be B, F, J, and D, because they all touch the very bottom. Okay, so I could ask you, you know, what will be older, E or G? Well, G is going to be older. And you're not going to have a question that's going to be, like F is kind of a hard one because it goes kind of all the way through. What's older, I or J? Well, J is a little bit older than I. But so you just kind of have to look at it, and I might draw, you know, a starfish here, and just that's not very good, but okay, so there's my starfish, and then here is my turtle. Okay, what's older, the starfish or the turtle? Well, A is older, and the starfish is in A, so it's going to be older, or lived during a, you know, older time period. So then you have radiometric dating. Y'all know I used to deal with radiation, and did X-ray, radiologic technology, and then MRI. And so I'm going to tell you all about radiation. So radiometric dating is the process that determines the approximate age of a fossil. And again, I said it's not like perfectly accurate. So what they're going to do is in the rock, they're going to measure the amount of radioactive isotopes in that particular rock or fossil. So how they do that is they know that radiation decays, and it decays based on something called half-life. So half-life is the amount of time it takes for it to decay by half. Okay, so using this we can kind of figure out when the organism lived. So for example, if I have 100 grams of technetium, which is a radioactive isotope, in this rock, I know in the half-life, technetium is usually 6 hours. So if the half-life is 6 hours, in 6 hours, I will have 50 grams of technetium. I should have put 6 here. This probably makes more sense. So if I have 100 grams when I start, in 6 hours, I will have 50 grams. And then in 12 hours, I will have 25 grams. And then I'll have 12.5 grams in 6 more hours, which is 18 hours. Okay? And then so on and so forth. Now, some things have half-lives of 1 or 2 million years. Okay, technetium has one of the shortest half-lives of all the radioactive isotopes, comparatively. Not the shortest, but it's one of the shortest. And so other things like uranium have a really long half-life. I don't remember the exact amount, but it takes a very long time for it to decay. And we know that the Earth was very radioactive in the early days. So they can go based off of these radioactive isotopes that are present in the rocks and use the half-life. So I know that if I have 12.5 grams now, that 6 hours ago I would have had 25. And 6 hours before that I would have had 50. So you can do the math either way. So this is just showing a radioactive carbon, how it decays. And I'm sorry, the picture didn't really show up very good. Okay, so we can also compare fossils. So we can compare fossils of 2 different species, a species that no longer exists, so one that is the same species, or 2 different species that maybe one's older and one's newer, or we can compare it to species that are living now. You can actually extract the DNA from fossils now. And so they can actually use fossilized DNA to compare DNA to currently living things. And they can also see if a gene mutation was present and if that is what caused speciation. So lots of great technology available. So punctuated equilibrium. I made mention of this before, but I didn't tell you the exact term for it. So I told you that if you have an environment that is very stable, then evolution is going to occur very slowly over a long period of time because the environment's not changing a lot. The organisms don't change that much. But then when you have a very much changing environment, when the environment changes a lot, that causes the organisms to change a lot and evolve a lot. So there's two ways that we describe that. Gradual evolution, which is just what it sounds like. Okay, so that's very gradual, very slow evolution. And then we have punctuated equilibrium. Okay, so punctuated equilibrium is very rapid evolution. Okay, so it's like they were same for a long period of time, and then all of a sudden, boom, lots of changes. And that, like I said, usually is related to the environment changing. So anyway, the rate of evolution is the same as the rate of speciation. Okay, remember, speciation is the formation of a new species. So as new species form, that is evolution. Okay, so the Cambrian explosion is one example of punctuated equilibrium where we saw rapid changes in the fossil record. So evolution again increases when we have lots of mutations, we have a rapidly changing environment, and we have small populations, especially when they're isolated. Now, gradual evolution or gradualism, like I said, that's going to happen more if the environment's not changing a lot and it's very stable. So this just shows you a picture of the difference between the two. So if you see here, gradualism, they're going to gradually change. Okay, very gradually change. Okay, with punctuated equilibrium, boom, boom, big changes, not gradual. Boom, 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 big changes. Well, this one stayed the same. Okay, but we had big changes here. Okay, so it wasn't gradual. We can't see how it slowly. But we got little differences here, and then a few more differences, and then a few more differences, and then a few more, and they add up over time. Okay, so. Okay, geologic time. Geologic time is basically starting from when they believe Earth began. So it's marked in MYA, million years ago, and they use this when talking about evolution. They believe Earth is 4.5 billion years old, and they think the first cells arose at 3.8 billion years ago. And you had simple unicellular organisms would have showed up in the fossils at 540 million years ago, and that was considered Precambrian time. And so they divided geologic time into three main basically time periods or eras. Okay, so the Paleozoic, Mesozoic, and Cenozoic, and then those are the eras are further divided into periods. So your three eras are Paleozoic, Mesozoic, Cenozoic, and that's not going to be on your test, but just to kind of understand it. And then each one of those eras also has periods in them. And then it does get further divided into that, but we're not getting into that. That's more Earth space science. But just for you to just kind of understand it. So there's been lots of, over time things become extinct. But there's been 5 major mass extinction events. And so, of course, one of those is dinosaurs. But they don't totally know what causes these. We don't have a time machine. But there's different theories about climate change. Could have, ice age or global warming, natural disasters, and asteroids. So those are some theories on what could cause these mass extinctions. Natural disasters, think like volcanoes and things like that. And if a volcano erupts, and it's a large enough volcano, it can cause massive damage to a large area. So they say if Yellowstone was to erupt, it would really, the whole United States pretty much would have, changes to the climate. So that very well could cause a mass extinction of animals or plants. And so this is, like I said, you got your major eras, and then they're broken down into periods. And so right now we are pretty much in the Cenozoic area. And each dotted line here shows a mass extinction. Okay, so this is when the dinosaurs were killed, and they were replaced by mammals, pretty much. And that's the most, I guess, well-known mass extinction. But there were others. Okay, so comparative anatomy. So anatomy basically means structure, okay, and we're comparing structures, okay? So you're looking for structural similarities. If they have structural similarities, like similar bones, they probably came from a common ancestor or shared a common ancestor, okay? So homology, homo means same. So homology indicates a common ancestor or shared ancestor, okay? So it's very important to understand if something is from a common ancestor or not. So there's 3 types of structures we talk about. Homologous structures is the first, so homo means same. So I want you to know that homologous structures are homo, same structures that have different functions. Function is the job. Okay, so a homologous structure is going to indicate a common evolutionary origin. Okay, that basically means that it probably came from a common ancestor or shares a common ancestor. Okay, divergent evolution can cause homologous structures. Okay, so they usually develop from the same tissues as embryos, and they might have the same structure on the inside, possibly look different on the outside, but their function is different. Okay, the primary example is what I showed you earlier. The arms and legs of different, like mammals, I guess you could say. So the forelimb, the arm of a bat, a human, crocodile, bird, they all have the same bones, but we use those bones for different functions. So bats use those bones to fly. Humans use our arms for lots of things. Crocodiles mainly use them for like running, walking, running, swimming, and of course birds are going to use it to fly too. So here's a picture of that, and they've color-coded it so you can see what bones they have in common. And because they have the same bones, they say that they probably came from the same ancestor. So that brings us to analogous structures. Okay, so analogous structures are the opposite of homologous structures. Okay, so they have different structures. Ooh, I can't spell different. Whoops, sorry. And apparently I can't get the eraser either. Okay, so they have different structures with a same or similar function. Okay, so a big example of this is going to be a bird and a butterfly wing. Birds' wings have bones in them. Butterflies, insects do not. Okay, so they have different structures, bones versus not bones, in their wings, but their wings are both performing the same function, to fly. Okay, so another thing would be like a fish with cartilage versus a fish without cartilage, or like a dolphin who has bones. Okay, so they have different structures, but their function is the same. Their fins would be to swim. Now, vestigial structures are structures that don't have any purpose anymore. And so they might find them useful in some species, but other species they're not. So like ostriches have wings, but they don't fly. And so they believe they've descended from birds, because even though the ostriches don't use them, maybe other organisms did. Cave-dwelling organisms that have eyes, and they don't need them. The pelvic bone or hind limb bone in whales, they don't need them. Okay, the appendix in humans is also considered a vestigial structure, so it has no function. So embryology, we briefly touched on this earlier. So embryology is looking at the development of embryos. So this is pretty simple. If you look here, we have a fish, a bird, a reptile, and a human. And because they have some similar structures and they look very similar at the early stage of development, the embryonic stage, they believe that they share a common ancestry because of that. And this is Over here is just another picture of embryos. Anatomical similarities, so things that share similar structures like a cerebrum, a cerebellum, a brainstem, that makes scientists believe that they probably came from a common ancestor. Biogeography. We talked about biogeography a little bit before. So we talked about basically how things are spread out on the earth. Okay, so this goes a little more in depth on it. So if you recall plate tectonics, we have large plates floating on the Earth's surface, and those plates are moving, which the movement of those plates causes the continents to drift slowly, extremely slowly. It also causes volcanic activity and earthquakes. So scientists do believe that all the continents were once one giant landmass called Pangaea, and over time they drifted apart into all the continents we have now. So biogeography is the study of how living things, plants and animals, are distributed all over the world. And they believe and they use biogeography to kind of decide how and when species evolved. So they've noticed that some species are only on certain continents. Like one example is apes are in Africa and Asia, elephants, marsupials are going to be in Australia. So they're seeing a trend there with where they are, where these animals were distributed. So this is just showing you Pangaea, what it would look like, and how each continent has specific organisms for specific places. Okay, so compared to biochemistry, so when we study biomolecules, biochemistry is kind of what you need to think about biomolecules. So this is a study, physiology is the study of processes and functions, okay? So scientists study different biochemical compounds, your DNA, your RNA, your proteins. They also look at the sequence homology, okay? So sequence homology are having similarities in your nucleotide sequences, like your DNA and RNA, or possibly amino acids of proteins. Okay, so if you have a similar sequence, so for example, A, G, G, C, T, T, A, if two organisms have, or they're very close and only have a couple letters off, that's sequence homology. Okay, homo is same, so it's similar sequences. Another thing would be having a similar protein sequences. So like the amino acids, serine, glycine, if you have these several amino acids in common or several proteins in common, okay? So if you have this particular sequence and you shared it with another organism, then that means they possibly could have came from the common ancestor. Okay, and this is just showing kind of like a phylogenetic tree, but it's also showing how you had new fossils that are going to show up and help look at that particular branch of the family tree, the phylogenetic tree. DNA comparisons, I mentioned before that DNA is the best way to determine if two organisms are related. Okay, so you can study the DNA and see that genes determine their characteristics, and then how the genes have changed or possibly mutated over time. So you can study the nucleotide sequences. So after they used something called gel electrophoresis, if y'all recall, we get the DNA bands. Okay, so that was all the lines. And then I can compare them to see if two things are related. Okay, so this is a red panda. Okay, this is, of course, a raccoon, and this is, of course, a panda. Okay, so they've done DNA studies, and as you can imagine, the red panda and the raccoon are closer related because of their DNA. But they call this a red panda. Okay, so DNA is going to be better to see if two things are related. Okay, so genes are not always in the same places on the same chromosomes if you're comparing DNA between different species. So another thing is different species have different numbers of chromosomes. So for fossils, it's easier to compare a physical similarity than DNA similarities for this reason. So this makes it hard to interpret and compare DNA when you have, an organism that has 46 chromosomes and an organism that has 1,046. So it makes it very difficult because we don't have a genome for every organism. We have a human genome. We know where the human genes are, but it would be very difficult to study DNA comparisons in multiple species, especially if they're not closely related. So that concludes this unit. So hopefully this was helpful. Don't forget to study your workbook, study your questions in your workbook. Make sure you know all of our scientists. And this is a very situational test. So I'll be giving you a situation a lot of the time, and I'll be asking you, okay, what is this talking about? Is it punctuated equilibrium, or is it directional selection? So a lot of what you'll be seeing on your test will be a situation that you have to then interpret. What are you seeing? So anyway, I hope this was helpful.